I need to wait for everybody to take the exam before I can release the scores, but hopefully I will be doing that soon. Um, so just be patient, please. But in the meantime, a couple of things. You have the lab exam on Thursday, remember, okay? So I think what we're gonna do is try to get you into a different room for that. Last year, one of the physiology labs on the bottom floor was available. That way, when you're done, you could go back to the lab if you wanted to keep working on the case study stuff. How is the case study stuff going? Making good progress? Okay. So we like to give you, because a 2-hour block, I don't know if you'll need a full 2 hours to take the lab exam. That way you could go back to the lab and not have to be quiet. No one else will be in there taking the exam. So I'm going to work with Duncan and Isabella. I meet with them today. So we'll try to figure that out and then send announcements of how we're going to handle that. Okay. And then we don't have any more exams in here until the final exam. So that is Monday of exam week. Really? Monday? Ugh. Monday of exam week at 2 o'clock. Ugh. All right. But we're going to continue on with this, and then I'm behind schedule. So I'm probably going to ask you to vote on what might be the other topics we'll cover. And I'd like to hear from you before I make that decision final. So after we finish the microbiome stuff, I'll look to see how much I can actually cover, and then we'll vote on it. So get prepared to weigh in on that. All right. We've been covering the microbiome. We kind of did a survey over the body. Now we're to the point where we can talk about the gastrointestinal tract. Some people will abbreviate that down to GIT for an acronym, or just gut is fine to say too. And they really, that area of your body is the most abundant in terms of microbes. Literally trillions of microbes reside in your gastrointestinal tract, all the way from your stomach to your colon. And so now, if you look at the progression, it's kind of interesting because the stomach has the fewest microbes. And then as you continue to work your way through the gastrointestinal tract, you get more and more and more. And so that's something I want you to pick up on. So with the idea of the stomach, probably a very good reason why there's not a lot of microbes there is the super acidic pH. And there really are only a handful of microbes that can adapt to that kind of environment and actually persist. And so here you're looking at Helicobacter pylori, which is famous for being able to be in your stomach environment. There are others. It's not just Helicobacter, but that's the one that's most famous. It can survive all the way down to pH 1, just fine. And it tends to kind of bury itself, so to speak, into the mucus lining of the stomach that your cells produce. You produce a mucus, and they can kind of hide away in that, and that does provide them some protection. And they're famous because they are now known to potentially cause ulcers. And it's not necessary that if you have this in your stomach, you'll get an ulcer. But they can show it. It's actually a very famous situation that I'll talk about in just a minute. Be aware that if you take medication, well, there's a couple things. People who are malnourished, this is typically in third world countries, you can get hypochlorhydria, which just means your stomach pH isn't as acidic as it should be. Here in America, and more advanced in terms of our economic and all of that, we tend to pop antacids if we have an upset stomach, and just be aware that that can raise your pH of your stomach temporarily, and that can set you up for potential issues of if a pathogen gets into you, it could then sneak past your stomach and not get killed because the acidity isn't strong enough, and it makes it down into your intestinal tract. There's been cases of Vibrio cholerae, where the epidemiologists and the doctors work with the people who are sick, and they find out that, in some cases, it's because they were popping antacids like it was candy, and they had their stomach acidity just wasn't acidic enough. So be careful about that. Okay. So Helicobacter has an interesting history because, well, ulcers in general, medical doctors thought ulcers were caused by spicy food or stress. And when my dad, who's 100% Finnish, and if you know anything about Finnish cuisine, it is not spicy. It is very meat and potatoes, fairly bland, traditionally. And so when he came home and said, my doctor said I have ulcers, we all laughed because he was a meat and potato guy. And so it was stress that he was under that was causing it. Certainly, but not the spicy food. And so we'll talk about how stress can impact your whole body and your microbiome. But it was Gary Marshall, Dr. Gary Marshall, and his colleague who actually won the Nobel Prize for discovering that it was Helicobacter that's actually causing the ulcer. Stress plays into that because it creates a situation in your body where Helicobacter can gain a foothold more easily. But he was onto it. He was a pioneer in this area of research, but at the time it was so entrenched. No, spicy food and stress, that's what causes ulcers. And so he got so discouraged by the pushback from other scientists that I think he was at a conference where he was on stage giving his presentation, and he said, I'll prove it to you. And he drank a broth culture of Helicobacter, drank it down, and said, watch, I'll prove it to you. I'm going to get ulcers. Would you do that? Oh my gosh. Talk about passionate for his field of science. He was just like, I know this is the case, but no one would believe him. So he drank it down, got ulcers. Luckily, it was sensitive to antibiotics because then he took antibiotics to kill it off. But he got the Nobel Prize. I don't know if we should award that behavior though. That's pretty intense, but they won the Nobel Prize for that. So if you want to read a little bit, you can read through this story about how he did that. But yeah, that's not something I would do. I mean, I love science. Microbes are cool, but I wouldn't go to that extent. Okay, so let's say we're past the stomach now. There's not a whole lot going on there in terms of who's there. But then we get into the small intestine next, and that is much higher concentrations of microbes per gram of feces compared to what we see in the stomach. They say, well, actually, this data is kind of like the whole intestine, not just the small intestine. This slide is about the whole intestine, okay? So there are way more anaerobes in your intestinal tract compared to facultative anaerobes, and certainly there's not many aerobes. All right, so your intestinal tract is a nicely anaerobic environment, mostly. And so facultative anaerobes and anaerobes, and particularly the anaerobes. So that makes it difficult to study because you need, if you're going to culture any of the microbes coming from the intestines, you need fancy equipment that can reduce, the oxygen environment, so that you've probably seen that on like TV shows where you have to put your hands into the, I don't know what they're called, but it's like a glove, and then you have the sterile flow hood, but everything's like there's no oxygen in there. And so you manipulate. It's very expensive. You need very special equipment. I think you might, I don't know if Isabella or Duncan showed you how much just an anaerobic jar costs when you did the anaerobic jar. Just that jar alone was, what was it, $700 or something just for a plastic jar. So you can imagine what some of this stuff costs. So early on, especially with babies who are newly born, probably the first thing the doctors did to you is give you a shot of vitamin E. They usually give it into your foot. And the reason they're doing that is at first you don't have any E. coli in your or in your intestines, and E. coli makes, did I say vitamin E? Vitamin K, I'm sorry. E. coli makes vitamin K for you, sorry. And you need that. And so they usually, as soon as the baby is born, I mean, literally, they'll say, is it okay if we give the baby a shot? And I think they still do that to this day because they know that E. coli is not quite there yet in terms of sheer numbers. And they want to make sure the baby has enough vitamin K. So E. coli is a facultative anaerobe, so it's capable of using oxygen, which if it's smart, it will do that. If oxygen's available, yep, I'm going to use it. Well, then that starts up the oxygen, but that's what allows other anaerobes then to come in, because especially those that are super sensitive to oxygen, if E. coli is doing its job of scarfing up all the oxygen, then it allows for other species to get in there and actually persist. So in general, you often hear this kind of quote, 1,500 different species hang out in your intestines. You do have some methanogens in there, at least a good percentage of the population, adult population, a few archaea, but yeah, lots and lots of diversity. So if it's been a while or if you've not taken a human anatomy, this is the general idea that we're thinking about. And when you don't need to memorize who's where and all of that, other than no helicobacters in the stomach, but this is just, and certainly this is not, 1,500 different species being listed here, but what I want you to pay attention to here is the shift in pH as you work your way down. Got it. The shift in pH, so stomach, very acidic, and then it gets less and less acidic as you work your way through the intestinal tract. You don't need to know, the sub areas, but know that the stomach's connected to the small intestine, to the large intestine, which is often referred to as the colon. And the other thing they're trying to show here, which is kind of implied, is that you have more and more microbes showing up as you move south, so to speak, through the intestinal tract. Okay. I'll show you another picture in a second here that gets at that a little bit better. Here's, if you've not thought about how your intestinal tract works, I'm not going to get into the huge physiology of it, but I thought this was a nice kind of overview of what's going on when you eat. And then the digestion, secretion and digestion going on. And then you start to see in the small intestine that the nutrients will get absorbed and get into the blood system. And then eventually the nutrients, after they're fully extracted and transferred into the blood system, what's left over goes into the large intestine and the colon and will be excreted out or defecated out. So we need to think about this idea that, your blood vessel system is busy taking those nutrients and delivering it to various parts of your body. And that is something we need to think about with regard to what are the microbes doing in your gut and how might they be impacting other areas besides your gut. So we'll come back to this idea a little bit. Here's another image of the gastrointestinal tract and with some other parts in there too. And they do a little bit better job here of explaining that, the microbe numbers go up. You get more and more per gram as you go down. And then you see the large intestine definitely has the highest population of microbes of the whole intestinal tract. And then they do show the appendix there too. Little appendix. So there's a lot of hypotheses, or there's been a lot of talk about what good is the appendix. I thought this was an interesting hypothesis. I don't know if anyone else has heard of any other ideas, but this one they're saying maybe it's like a refuge for microbes to hang out, so that if there's a washout of microbes in the rest of your intestinal tract, they can recolonize. So it's kind of like a little reserve bank of microbes in the appendix. Has anyone heard of that before? I don't know if there's any other hypotheses out there, but that, to me, that made sense. Some doctors think the appendix is useless. But I think this makes sense that there is a use for it. Evolutionarily speaking, there should be a use for it. Okay, this is an awesome picture. This is in a mouse intestine, but this is such a nice visual. And this I want you to think about a lot, because what we see here is also what happens in the human body. So the blue, that's the mouse cells, okay? All of the colors in the upper left here, all of that, those are microbes. And then the green separating, that's the mucus of the intestinal tract, the mucus layers. And I don't know how well you can see it with the lighting in this room, but you do see, especially in this area here, that layer of mucus, you do see, if I erase this now, you do see some cells in there. Okay, there's some red cells here, like a green cell there. Okay, so those are microbes hanging out in the outer layer of the mucus. That's a really important point here. So when you look at the mucus layers, and they've done most of the research on the colon, because that's where the most microbes hang out. So they're very interested in that area. So you have an outer layer, that's the looser layer that we were just looking at, where some microbes were hanging out. The inner layer is denser, and that's the layer that's stuck right to your epithelial cells. And it's much denser. The drawing down there, I think, is a nice visual to get across that point. And then the outer layer that's in close contact with the microbes in your intestinal tract, that's looser, and they're trying to show that bacteria can hang out in there. All right. They have recently discovered that both layers actually have a lot of bacteriophages in both layers, and that they are busy infecting bacteria. And so they think that maybe these bacteriophages are doing us a service by making sure that there's not an overpopulation of bacteria that close to our epithelial cells. So that kind of makes sense, because it's this, you're a superorganism, you're symbiotic with trillions of microbes, but you need to keep a good balance with your microbes. You don't want them getting crossing boundaries they shouldn't cross, which could then set up issues for the human body. So it does make a lot of sense to me when I read that bacteriophages are there and they're helping to keep population levels at appropriate levels, so everything stays in balance. So they're referring to it as non-host derived, because it's not part of your immune system. It's just phages there providing antimicrobial defense. So another good reason viruses can be useful. Okay, so here is a visual trying to get at this idea of the mucus in the stomach. So the adherent mucus, that's the dense layer, you can see it's dark gray, and then the lighter gray is the non-adherent, that's the looser layer. And remember, the small intestine has a lot of those villi that create great surface area of the epithelial cells in the small intestine. And they're trying to show that sometimes the mucus is thick enough where the whole villi are covered over here on the right, that part. And then sometimes they're saying, sometimes the mucus is not. So this part they're trying to show, yep, all the villi are covered in the small intestine, but here they're showing, the mucus isn't quite thick enough. So you have to think about those types of issues going on in the different regions. And then you can see in the colon that there's quite a nice thick layer of the outer layer of the mucus. And that's where the microbes like to hang out. Many microbes like to hang out. Not everyone hangs out there, but. So also be aware that there are key microbes that hang out with you on a regular basis that like to eat the mucus. And that's not necessarily a bad thing. It actually, they're finding, so this was really confusing earlier on, like a decade ago when they were discovering this. They just assumed all microbes that eat mucus are bad. They're doing harm to the host. And if you go back into that literature, it gets very confusing. So don't do that. Go into the newest literature where they're now saying, oh, wait, they're actually helping your gastrointestinal tract. It kind of, because you keep making mucus, right? Continually, you're making mucus. And so that could really build up. And so the idea that there's some microbes there to eat the mucus, keep the levels adequate, but not too, thick. And they are also finding as the microbes are eating the mucus, they're actually stimulating your immune system cells in that area, and it's a good thing. So, but initially, if you go into the older literature, it's confusing. However, if you don't eat well enough, a healthy enough diet, you can create an imbalance where the microbes that are eating the mucus are just don't have enough other good stuff to eat. It's not like they only eat the mucus, they eat other stuff too that comes from what you eat. But if your diet isn't the greatest, then they're kind of left with no other resources, so they just keep eating your mucus. They don't have other nutrients coming in that they can take advantage of. And what they're finding is that if you have low fiber diets, it really seems to create this situation where the mucus eating microbes eat too much of your mucus. And then that causes you problems. Does that make sense? Okay, that is really important to think about here. Yes.
When they talk about eating fiber for like the health, is it, I guess this might be silly, but I was under the impression that we're not like actually using any of the nutrients for fiber. Is it just physically like helping the tract?
There's different kinds of fiber. Like there's some fiber that's more the physical that just help keep you from not being constipated all the time. There's that kind of idea. But then there's fiber that is not really something your enzymes work on, but the microbes do. And we're going to get into that quite a bit because that's one great way to promote the good microbes in you by giving them the types of fiber that they need. So we're going to talk about that. Here's a visual. They're trying to show if you don't have enough fiber in your diet to feed these microbes that hang out in the mucus. So fiber free, they're trying to show how the mucus gets overeaten, overgrazed. And now they're showing a breakdown in the epithelial layer of your intestinal tract. That's not good. Right? We don't want that. And so if we have high enough fiber in your diet, the mucus layer stays at the right levels. You don't get overgrazing of the mucus. Microbes don't have a chance to get, cross boundaries they shouldn't be crossing and starting to cause problems. So I thought that was a nice visual to show you that idea. Oh, there's more to the key here. Who's what here? So the code here. So they're trying to show, everything's in balance on the left with that teeter-totter idea. They're saying, yep, everything's in balance, everything's good with the high fiber or the correct fiber diet. Here you're seeing an imbalance. So when I listened to a podcast of a microbiome researcher and the parting question the interviewer asked is, what is one thing you can tell people listening? What's one thing they can do to really support their microbiome? Can you guess? He said, eat your fiber. He says that's a simple thing to do, doesn't need fancy prescriptions, and you're not spending a ton of money. So he said, eat your fiber. That's really one of the biggest things you can do. Okay? And I agree. So we'll talk a little bit more about that. Here's another picture trying to map out the intestinal tract area. And there's some stuff on here you don't have to get into in huge detail, but you do see that mucus layer again. So your gastrointestinal tract is one of your major areas of your body where your immune system cells are there. Your immune system have to mature in this area, and the reason or the ways in which it can mature correctly is through the interactions with microbes that coexist with you. So it's like the microbes there need to kind of work out your immune system so that the immune system gets challenged, but learns to tell friend from foe. You know, all of that's going on as you're developing and growing from, you know, baby on to adult. So microbiota is very important for that. The IgA and defenses, that's immune, dendritic cell, lymphocytes, so that's all immune system stuff going on. And then this middle section here, that's the epithelial cells, okay? And what they're trying to show here is, you know, you have the neighboring epithelial cells kind of connected, so to speak, by these tight junctions. I want you to know that phrase, please, tight junctions. Those are the tight junctions that are keeping neighboring cells together, and so that there's no easy gap in between. As you can imagine, if the mucus gets down, overgrazed enough. Let me go back maybe to this picture, or actually, well, let's go back to this one. So if the mucus gets down too far, they're trying to show here that the gap junctions are starting, the tight junctions are starting to get degraded. And then that can allow either microbes to get through or microbial products to get through. And then it can get into your blood vessel. It can over trigger your immune system. So I'm going to be referring to tight junctions a fair bit in the sense of if that barrier gets compromised, that's when you can start seeing some issues, especially what they call inflammation in that area. Here they're showing if things get a little wonky, and they're trying to show that you have some kind of dysbiosis. So that word dysbiosis, I want you to know that just means there's an imbalance. It's off the normal, and usually in a bad way. So here they're showing that the mucus is getting altered. It's getting a little thinner than the previous image. And then they say, well, then you have a chance where pathogens might be able to persist a little longer. You might get some toxins being produced that could get through the tight junctions if you have your mucus layer too thinned out. And then they're trying to show that these endotoxins that microbes make can get all the way through and get down, you know, into the blood system, and then can start coursing through your body to different sites, right? So things that go wrong in terms of with the microbes in your gut can have far-reaching impacts on your body. So you have not only the immune system in the gut getting over-triggered or triggered inappropriately, but then some of these chemicals can get into the blood and travel. So when they started learning that this was happening and that the mucus layer is so critical, and then they did a lot of mouse studies where they were looking at, you know, what happens if it gets thinned out, and they started piecing this all together. Basically, if all of this is happening where you get pathogens gaining a foothold, you get toxins getting into your blood, over triggering your immune system, basically what starts to happen is chronic inflammation, okay? Chronic inflammation. So this is a book written by Paul Clayton. He came here several years ago to give a seminar on the microbiome, really good seminar. He's a physician, MD, PhD, I believe, over in England. And the title of his book is Out of the Fire, and then the subtitle is Why Chronic Inflammation is the Root of All Disease and How to Put Out the Flames. And I think that's a catchy little statement there. That's pretty much on point. And so he is pretty much saying that if your body is in chronic inflammation, that's the start of all kinds of potential disease impacts that can lead to a lot of different diseases, whether it's obesity, heart disease, cancer, immune system issues. And so, you know, he's making the point too, that a lot of drugs that you take, it's only dealing with the symptoms. It's not dealing with the cause. And so you're learning how, you know, the drugs give you maybe a better quality of life in terms of the symptoms might go down, but it's not really solving anything in the sense of it's not going to the actual root of the disease, what's really happening. And so with the most recent research coming out in the last 5 years or so, what they're finding is there's several things that can lead to chronic inflammation. Genetics play a role. Your genetics that you inherited from your mom and dad does play a role, but not as big as they thought. And they're realizing environmental impacts has a really strong impact, especially what food choices you make, because that's your most intimate experience with your environment is what do you eat and take into your body. The mucus layer, loss of that barrier is a problem. A lot of people refer to that as leaky gut out in the mainstream media. Hyper-belligerent immune system. That just pretty much is saying your immune system gets triggered and kind of turned on and there's no off switch. And it leads to inappropriate immune system functioning. And then dysbiosis, they're saying, is also playing a role in chronic inflammation. So I'm talking about the inflammation that you can't feel per se. Like if you cut your finger, like a paper cut, you know how swollen and painful that can get, just a tiny little paper cut. That's inflammation. And that's actually good because it's delivering white blood cells and nutrients to the cut. And that you can feel because it's very painful. I'm talking about systemic inflammation of your whole body often. It can be a whole systemic thing, but you don't feel it per se. And so that's what they're getting at here. It's that chronic low-level inflammation that you don't really perceive. And that's what's leading to a multitude of different diseases. So this is another visual trying to get at that, where they're saying your environment, a lot of what they have here is, you know, what kinds of drugs are you taking, what foods are you eating, and all of that has an impact on your microbiome. And they're saying, what if you do that inappropriately and you get an imbalance? Okay. And so they're doing the teeter-totter thing here where they're showing, in this case, they were looking at two groups of bacteria, the Bacteroidetes and the Firmicutes. And they're saying, you know, if you create a situation where those get out of balance with each other, you can get issues down the line, including obesity, heart disease, and so on. Here's another image where they tie in the inflammation idea. Okay, so here they're showing the intestinal epithelial cells right there in that. And they're saying, you know, you're eating all these foods. And in the initial image right here, they're showing your gram negatives and your gram positives look like they're in balance. But then who's winning in the imbalance when the teeter-totter starts to teeter? Is it the gram negatives or the gram positives going up? Yeah, gram negatives. So key in on that. The gram negatives are increasing. And they're finding what happens is, remember now, gram negatives have that outer membrane, including the lipid A portion. All those yellow boxes are depicting the lipid A. That's what our body responds to. We can actually go into shock if we get too much of that all at once in our body. So the LPS, okay, they're saying the LPS can leak through if you have a leaky gut. You can get the lipid A portion in particular getting into your bloodstream. That's the endotoxin idea they were showing in the previous. So they're saying if you have too many gram negatives, you might have too much endotoxin or lipid A getting in to your blood system. And here, you know, they have the inflammation and the big red blow up there. And so again, they're showing an imbalance of the microbes leading to a systemic inflammation over time. So they're starting to piece this together with a little bit more detail. And so with some of the review papers I was reading, they're saying, yeah, that in the mucus in your intestine, it really is a nice shield. It's very protective for you. It's what, you're making it and constantly and replenishing it. And it's good if it's nice and thick because then the endotoxin or the lipid A can't get through. But if you have inflammation going on, low fiber diets, again, they mentioned, high alcohol consumption, high fat diets, and some other factors, then the mucus decreases, the quality isn't there, and you get more of a leaky gut situation. And so the lipopolysaccharide or the lipid A in particular can be a toxin, and that can start to negatively impact your health. You don't need to know how it does it, but if you're curious, all the writing gets into that a little bit. Yeah, I'll get into some different diets that are popular right now. Like they talk about the ketogenic diet, that's high protein. We'll look at some of that too. From what I know, I'm not an expert in like the ketogenic diet. I know it's high protein. And I know that people can lose weight on that. But to me, I would feel uncomfortable myself, always having that diet. I could see doing it for a while to lose weight maybe, but I think it kind of messes with your microbiome. It's not a very natural human diet, at least evolutionarily speaking, when you think about how we evolved to eat. I'm not sure that's really, and this is my opinion, okay? That's just, I wouldn't follow it myself long term. I've never tried it either, but I've heard it can cause nice weight loss if that's an issue. So this is another visual trying to get you to think about this idea of all in purple is like all the things that could potentially be impacting humans. And the upshot is that it leads to a dysbiosis and altered microbiota, which then if the mucus gets degraded too much, you get damage to the tight junctions, the epithelial layer of your intestines. You start to get overreactive immune response, which leads to chronic inflammation. And then that leads into other disease states. And so they're really starting to piece this together a little bit better, looking at, boy, it's all coming down to this chronic inflammation is really what's causing, depending on your particular situation, what you ate or what pathogens might be in you, but you get that chronic inflammation and then boom, you're off into leading into a disease state. So I want to talk a little bit more about that in the next slide set. I think that's the end of this slide set. So this slide set's kind of getting, the one we just covered is looking at kind of all the different body areas and ending here now with the gastrointestinal tract. But now I want to get in a little bit more on what are the gut microbes doing and how are they impacting different areas of your body. But before we get into that, I wanted to talk to you a little bit about how are they actually doing this kind of research to get the data that I'm talking with you about. Because as you know, in science, if you have faulty data or poor experimental design, how well can you trust the results and conclusions? So let's think about that a little bit. If we're thinking about the human microbiome, it's not like there's people lining up to get surgery done on themselves so you can get sampled, right? I mean, that would be the way to sample, especially your small intestine. for microbes, they'd have to cut you open and get in there and take samples. Not many studies will allow for that. So it's fecal samples you work with. If they're doing a human study, that's primarily what they're doing. With mice or rats, on the other hand, they can be doing fecal samples, and that's what most of them are. But some scientists will do a little bit more rigorous if they want to know more specific data, get data on, you know, like, where are these microbes? Are they in the small intestine? Are they in the large intestine? And if that's an important aspect of their research, then they would have to do surgery in order to take these samples. There's now lab created organoids that they refer to, organoids. I had to look that up. Like, what are they talking about? The jar or the nickname is mini guts. And so they actually will harvest, like during colonoscopies or biopsies where they have to go into the patient, they'll actually harvest cells and grow these organoids in the lab. And then they start doing the research on it. And I thought, well, that's great, because that certainly, if they had to go in there anyways to do a colonoscopy, they can sample and then do all these experiments in the lab. So when they grow human cells, there is like a nutritional broth that they usually grow the cells, like, it's kind of like that blue bottle idea, like a thin layer of nutrients, you know, just a little. And then it's like, when I did it, I grew cancer cells when I was doing research over the summer one time, and it was Petri plates with a thin layer of nutrient broth, and then the human cancer cells would grow in like a mat over that. And yeah, and so I'm guessing it's something like that. I've not seen one with my own eyes, but I've read some papers where they worked with mini guts to generate the data. So let's say they take their sample, and then it's like, okay, what do you do with it? It's typically DNA analyses. They want to know, they want to do large metagenomic sequencing, where they could take a fecal sample and send it off and say, sequence all the genes in this sample and then tell me who's there. And so there's huge, you know, powerful computers that can assemble all this data with databases to compare it to. And so you often hear the term metagenomics. We did metagenomics on the, you know, fecal samples, or we did shotgun sequencing. And those types of studies are great for telling you who's there, like all the different species, who's here, you know, and that's can be super important. Then you might want to choose to also do, or maybe just do, RNA analysis. If you got enough money, you'll do both. But RNA analysis is interesting. They're doing more and more of that, which I'm very excited to see. So you might hear the term metatranscriptomics, big mouthful. Functional activity. So if you're sequencing the RNA, that's telling you, at least when you took that sample, what genes are turned on. Okay, that's huge. If you can get at what are they doing, what are they functionally doing when you took that sample, that's huge, especially when you get down in the microbial world, because as you know, microbes are great at horizontal gene transfer. So, you know, it's kind of hard, like knowing who's there can be important, but sometimes it's kind of irrelevant sometimes, because they do so much horizontal gene transfer. It's hard sometimes to even tell if they're different species, because they've got so much of that going on. So very often I'm very interested in what are they doing, not so much who's there, because a lot of microbes are all doing the same thing. Maybe some microbe only does something very particular, but knowing all of that is very, very important for how are they living their life, and how is that going to impact the human host. And then this diagram down here is trying to get at, you often hear about alpha diversity and beta diversity. And so in this case, like alpha diversity would be like one human patient that they're sampling, or I've even seen alpha diversity. Well, beta diversity here, they're showing two different hosts would be beta diversity when you're comparing that. But I've also seen beta diversity described as like when one of Dr. Lafferty's students was doing her seminar presentation. She was looking at what's happening in the small intestine of the bear versus the large intestine of the bear. And that is beta diversity because they're very distinct areas of the intestinal tract. So, but anyways, if you hear alpha and beta diversity, that's what they're trying to get at here with those terms. Okay, so the other thing you should be aware of whenever you read these papers is that it really does depend, how did they sample? So they often say, what's the sampling effort? And then how deep did they go in the DNA analysis? So if you sent off a sample and say, tell me who's here, but only go to the family level, or tell me who's here, but go all the way to the species level, that's different resolution, okay? And so they talk about shallow sequencing, which would be like, go to the class level and we're good. Or go all the way to the genus level. That would be more deep sequencing. And so even just, how do you take the sample? What do you do with it? Do you refrigerate it? How quickly was it acted on? All of that you should be looking at when you read these papers. And then look at, well, what method did they use to extract the DNA? That matters. And how did they prepare the library, so to speak? And you can get all that information from the companies that do these different protocols. And then depending on how they did this, you might miss small populations that are hanging out with the host. And initially they thought, yeah, what's the big deal? So what? Now they're realizing that the rare populations or the rare species does not mean they're unimportant like they used to think. And so if you just think about the keystone species idea that you hear about in your ecology classes, where, you can have wolves, very few in number out west, and yet they have a really strong impact on the ecosystem of, hunting down the caribou or the deer and how that changes the whole ecosystem. So a wolf would be a keystone species. Well, there's keystone species they're finding in the gut that are also playing, they're small in number, but they're mighty. They're able to have and a disproportionate impact on the human host, even though there's very few of them. Then there's germ-free mice. They do a lot of experiments with germ-free. Often they'll compare germ-free mice versus conventional mice in these studies. They'll do human fecal sample comparisons between different humans. They'll look at differences and similarities. Some really nice studies done on human identical twins, where, at least the genetics are a variable that can be kind of wiped out because it's, well, kind of, because they're identical. So that's kind of a gold standard. Fecal microbiota transplants is also becoming much more popular for doing some of these experiments. And they very often take fecal samples from humans and put them into germ-free mice and then watch to see what will happen. And then right now, there's a lot of people, a lot of scientists saying we need longitudinal studies now more than ever, but that requires a big grant because what you want to do is sample the patient over time. So starting often when they're young, when they're kids, and keep them in this test until they're adults and, or taking samples essentially before, during, and after any onset of any disease so that you can track what's happening in that patient as, if a disease pops up, you can see what's happening before, during, and after. And that's really, there are some longitudinal studies in progress right now. But that's, this last bullet point with the longitudinal studies, that's kind of like the gold standard. A lot of scientists say if we could do that, we would be, get some great data to work with and some really powerful data to draw some good conclusions on. So, okay, so let's stop there. We ran over just by a minute. Thank you for your patience, and we'll continue on with this on Wednesday.